This protocol is significant because it can be used to interrogate the immune repertoires of human donors to identify rare antigen-specific B cells. This technique allows us to obtain natively paired heavy and light chains, which can be rapidly cloned, and then directly tested against the antigen or antigens of interest. Demonstrating the procedure will be Mike Morton, a technician from our laboratory.
At least 1 hour after thawing and recovery, collect the donor PBMC by centrifugation and resuspend the cells in 50 milliliters of ice-cold max buffer for counting. Pellet the cells by another centrifugation and isolate the CD22 positive B cells by positive selection with CD22 microbeads according to standard protocols. Collect the bead-isolated CD22 positive cells by centrifugation and resuspend the cells at 4 × 10⁷ cells per milliliter of FAX buffer concentration. Add an IgG CD19 CD27 antibody cocktail to the cells according to the manufacturer's dilution recommendations with gentle mixing, and aliquot 10⁷ cells into a 1.5 milliliter microcentrifuge tube for the negative control. Add dual-labeled biotin streptavidin tetramers to the negative control tube at a final concentration of 36 nanomolar each for the PE and APC multiplied by the number of peptides in the sort tube. Bring the final volume in each tube to 0.5 milliliters with fresh FAX buffer, and add the peptide tetramers to the tube sort at 36 nanomolar each for PE and APC.